Welcome again to our SNAC seminar. I'm really excited today to have Emily Finn join us from Dartmouth University. I got the pleasure of meeting Emily a few years ago at a WISPR conference for brain function. And that one of my favorite types of conferences because everyone goes there and has meetings in the morning and then they just spend the whole day hanging out or going skiing or snowboarding and then meet up in the evenings to have more chats. And so Emily and I both got to hang out there and had some really fascinating conversations about thinking about brain networks and network reconfiguration and cognitive function and all these other really fun things that I think are at the kind of intersection of cognitive neuroscience and network neuroscience at the moment. And then after that, I was very fortunate that Emily and Monica Rosenberg, one of Emily's collaborators, invited me to come speak at HBM about some stuff, and they very kindly put me in their very popular symposium where I got up and sort of hand waved all about noradrenaline when they were actually doing really cool, sophisticated connectivity work. And so I've been watching Emily's career as it blossoms. It's been really fun to watch this kind of really exciting combination of kind of creative cognitive neuroscience questions, but also really sophisticated imaging analyses. And I really love the kind of direction that Emily's pushing things towards sort of naturalistic processing, people watching movies to really take advantage of kind of cognition in the wild, as it were. But also, really excited to see some of the layer fMRI work that she did with Peter Bandettini and colleagues that is kind of pushing the precision of fMRI in a way that I think we really need to if we want to start taking these ideas from computational models and mapping them onto brain imaging analysis so we can really kind of wed the two together. We need that precision to be able to do that properly. So really excited to have you here today, Emily. Thank you so much for joining. If you want to share your screen, and then looking forward to hearing you talk.
Sounds great. Thank you so much, Mac, for that way overly kind introduction. I feel like the stuff you guys are doing is like incredibly sophisticated these days, and yeah, so that's very flattering of you to say. And I'm excited to be talking to this group, and not having the in-person conferences, I just really miss sort of seeing people and catching up with people. So it's great to have the opportunity to do that over Zoom. So I should preface this with saying this is actually, I was telling Mac this before, this is actually like a much less formal talk than I usually give. And I'm hoping that, people want to just chime in, and I'll try to monitor the chat. If I don't see something, just like unmute and shout at me. But I know you guys were interested in the layer stuff, which is really interesting. And I hadn't actually really given a talk on that in a while. And rather than sort of try to mash up my naturalistic spiel and my layer spiel and pretend that there's a strong relationship there, I decided I would just sort of give two different talks. So I'm going to start with the layer stuff. And so then there'll be kind of a natural break before the naturalistic stuff. So if you're more interested in the naturalistic stuff, I guess feel free to leave and come back at some unspecified time or tune out for a little bit. Yeah, so I'll just kind of dive right into the layer dependent stuff. And I should say strongly upfront that this was work done that I did as a postdoc with Peter Bandettini, as Mac mentioned. And it was done in very strong collaboration with the second author on this paper, Renzo Huber, who really is sort of the physics mastermind behind pushing this technique forward. So to the extent that I can't answer certain questions, I will refer you to Renzo. He'd actually be a great guest, I think, to have if you guys are interested in learning even more about this. But I can kind of give you the flavor and the perspective of where I think this is at in terms of looking at sort of higher order cognitive applications. So I'm just going to start by kind of, oh, sorry, I heard an echo of myself, but oh no, it's gone. Okay. So I'm going to present the sort of main study that we did on this a couple of years ago now, actually, and then also talk about some of the kind of newer work that mostly Renzo's been leading, but I've been sort of involved with, and talk about some of the challenges with doing layer fMRI specifically in higher order association cortices. And so if people have questions or want to ask, want to talk more about that, that would be a lot of fun. But to start with this first study, so our motivation for this study, we had sort of two sides of motivation. The first was a theoretical motivation. And so we can sort of break down this idea of working memory into a series of functions. We have to encode a stimulus, then we have to maintain some representation of that stimulus over the course of a delay period. And then we have to use that maintained trace, so to speak, to make a memory-guided behavioral response. And so this function broadly has been linked to dorsolateral prefrontal cortex in both humans and macaques. And interestingly, there's both some sort of older classic work now and more recent work from the monkey side suggesting that these sort of subfunctions, so by that I mean the cue, the delay, and the response, might be localized to distinct cortical layers. And specifically, findings from Pat Goldman and Rakesh back in the late 80s and early 90s, we're finding that a lot of cells that were most sensitive to the cue itself, the thing to be encoded, were to be found in sort of the more superficial layers, specifically layers 1 and 2, while the cells that were most active during the delay period, while the animal was trying to maintain the trace of that location, tended to be located in sort of layer 3 and specifically deep layer 3. And then finally, when it came time to actually make a motor response, it seemed that the most responsive cells were in deeper layers, predominantly layer 5. And so there's been sort of evidence since then. Those were the first few classic papers kind of suggesting that, but it had yet to be shown in humans. And so we thought that this was a really good kind of test bed to see if we could expand some of the layer specific techniques to a more cognitive task in a quote unquote higher order association region of the brain. And so that sort of brings me to our methodological motivation for this, which feels, I mean, this space is moving so quickly now. So there have been more studies since ours, but at the time there really hadn't been much venturing into territory beyond primary cortex with these techniques. So we really wanted to see if we could get this to work. And several advances in fMRI in the years leading up to this project sort of made this possible. So as I'm sure many of you are familiar with, so increasing field strengths, more sensitive contrast mechanisms, which I'll talk a little bit about, and also better analysis methods had been allowing investigators to achieve this so-called layer-specific functional resolution. And I'll come back to why layer is in quotes, but you can probably guess it's not always perfectly matched to cytoarchitectonic layers, but it is sort of depth dependent in a way that lets us infer presence of activity in what we think are certain cortical layers. So when we did this project, as I mentioned, there had been some really beautiful work using these techniques in primary cortex, like primary auditory, primary visual, motor, and somatosensory, but there hadn't really been much up here, for example, in prefrontal cortex. So we wanted to see if we could develop and apply layer fMRI tools to show layer-specific activity in human DLPFC during different periods of a working memory task. And to do this, we borrowed sort of a very classic task where we had sort of two different conditions according to whether people had to simply maintain information in memory or actually manipulate that information. And so what this looked like, it was this string of random letters, essentially. So this was what people had to encode. They saw this for about 2.5 seconds, and then it disappeared for just a moment. And then they got a cue telling them whether they needed to alphabetize those letters. So in other words, rearrange them into alphabetical order in their minds, or just simply remember them in the order that they saw them. So they don't know which task they're going to get when they see the stimulus. So they have to sort of encode and remember the stimulus. And then they're told to either alphabetize or remember it. They have about 9 seconds of a delay period. And then they get a probe letter, and their task is to make a button press response indicating where that letter would appear in the order. So in this particular example, if you were told to remember these letters in their normal order, the answer to this would be 3, and so you would press the 3rd button. If you were told to alphabetize them, the answer would be 2, so you would press the number 2. And so the reason we sort of beefed this up is there was some evidence the evidence in humans was a little bit mixed. So simply maintaining a string of letters like this, or a string of, I guess they don't use letters so much in monkeys, but maintenance certainly is enough to sort of trigger DLPFC in monkeys. There's some evidence that in humans that's not quite hard enough, and you have to, you get much more robust PFC activation when you actually have to manipulate information held in working memory. So we sort of added this contrast here to tease that out. And then we had a second type of trial that we are calling action versus non-action. So in this trial type, all of these trials were alphabetized trials, although people didn't necessarily know that they were in this, oh no, sorry, that's not true. They did know this. I'll come to the run structure later. But essentially these trials were identical up until the moment of the probe. So people are still having to encode the stimulus and maintain it over the course of this delay. And then there are some like catch trials, or like kind of the opposite of a catch trial, where it ends up that you don't actually have to make a response. So there's no button press response. You've still done the working memory bit, but you don't actually have to, you don't get a probe letter that you actually have to respond to. And so—
Can I ask real quick, Emily?
Yeah.
Has anyone done like whole brain imaging of that first task, the like maintain versus manipulate? Because I mean, I know Despo had that paper back in the 90s, but it seems like working memory manipulation often just gets kind of called a synonymous with something like mental rotation, and that's about as far as people go. But I'm just curious whether there are data sets out there and whether you guys did any like sort of scans with the whole brain rather than just in the DLPFC.
Yeah, that's a good question. I mean, a lot of the, like, I feel like this is a paradigm that's really popular in like, yeah, like the late 90s and early 2000s kind of thing. So there's a lot of sort of classic work from back then, which is where we got our inspiration. So yeah, this paper here, you know, showed that DLPFC was the main region that gets recruited above and beyond, you know, for manipulation over maintenance, at least in humans. And then there was some additional lesion work in humans suggesting that people with lesions to DLPFC don't actually show deficits in maintenance. They only show deficits in manipulation. And so, but yeah, so we did a little bit of piloting of this task at 3T, but not like with the whole brain, but not in a like super formal way. So
yeah.
And there could be literature out there that I'm not aware of, but yeah, it might be kind of like ripe for an update.
Yeah, cool. Thanks.
So, yeah. So, right, so then to walk you through our hypotheses more specifically, we had basically hypotheses about which layer we would see activity in and when, according to these different trial types. So we hypothesized that in the upper layers, we would see activity that kind of rises once you see the stimulus and it stays high over the course of this delay period, specifically in these alphabetize or manipulate trials. Whereas in trials where you're simply asked to just remember or maintain the information, we might see an initial activity as things get encoded, but then once you see that cue telling you that you only have to maintain, we expect to see a difference, basically lower activation compared to the manipulation condition specifically in the upper layers. We hypothesize that this is due to these recurrent connections between these pyramidal cells in deep layer 3 that have been the focus of the monkey work and that are believed to support in some way the maintenance or the manipulation in humans of this information. So in deeper layers, we hypothesize that you know, these might rise a little bit once you get the cue and during the delay period, but consistent with the monkey literature, we expected to see the strongest activation in deeper layers once it actually came time to make a motor response. And specifically in the trials that called for an actual action, so where you see a probe trial or you see a probe letter and have to sort of convert that memory trace into a decision. Whereas in the non-action trials where people don't actually have to make a response, we don't expect to see that peak in activity. This we sort of hypothesize would be due both to some recurrent activity sort of within the column, but then also input and output sort of communication with premotor systems as people prepare to make that response. So this was our hypothesis. And I'll talk a little bit about the acquisition itself. So this was done on a 7T scanner, Siemens at NIH. We had 15 subjects that came for 20 sessions total. The main contrast mechanism that we were using was this sort of innovative, he's shared it with like the entire world now, so lots of people are doing it, but this is sort of Renzo's baby. So it basically, there's two near simultaneous contrasts that we're acquiring at the same time. So we're acquiring, you know, your good old bold signal, and that this tends to be sensitive in the sense that you need less activity, I guess, to evoke a strong signal, but it's less spatially specific, and that's because of the sort of draining vein issue that can bias you to see activity in superficial depths that you might misattribute to more superficial depths, but that's actually coming from deeper layers. So we actually interleave, every single bold image is interleaved with a vaso image, and vaso, for those not familiar, stands for vascular space occupancy. And this is a contrast that's less sensitive than bold, but it's much more spatially specific. So you can more directly interpret it's more sensitive, I guess, to activity in deeper layers, or there's less of a bias towards superficial depths with the vasocontrast. So this literally is an interleave acquisition. So every other TR is of a different contrast. When it comes time to analyze the images, we do sort them and sort of pre-process them somewhat separately, although we use bold to sort of correct phaso in certain ways that I can maybe go into detail about, but we'll probably just refer you to Renzo's papers. But basically by combining these two in real time, we kind of get the best of both worlds in the sense that we get more sensitivity so we can better localize the activity and find the sort of biggest hotspots, but then we can also be more confident about localizing it to specific cortical depths. Another slight innovation that we did for this study was we actually ran an online functional localizer. So as the subject was in the scanner, we had a sort of practice task run with a whole brain acquisition at standard resolution, and we did a real-time GLM while the subject was still in the scanner to select the ROI that we wanted to focus in on. And again, I should say, I mean, the technology has come so far, even in like the 2 or 3 years since we collected this data, I'm actually not even sure how necessary this would be anymore, although I think it would still help in a lot of cases. It's very difficult to optimize these sequences for a true whole brain acquisition. So you're always gonna have like areas where there's more dropout or ghosting, or you have sort of these really insidious artifacts that are just hard to avoid when you're pushing the resolution this high. So the importance of doing something like this was that we could kind of localize the area within a subject in real time and then position our slab in a way and take a few like dummy scans and see that, you know, we were actually getting good signal in the area that we most cared about. So it took a little extra time, but I think it was really worth it. And so the main protocol then for the layer specific imaging that our sort of workhorse protocol was this axial protocol. You can see the coverage here, it's pretty thin. So that was, you know, especially important to do this online localization to make sure that we weren't missing the hotspot, so to speak. And our TR here was 2 seconds. So 4 seconds in between images of the same contrast type. So that's a little bit slow, and I'll come back to that when I talk more about sort of the challenges I think that are still outstanding with this technique. And then later in the study, we came up with this even higher resolution protocol, which is the sagittal slab. And we use this primarily for visualizing layer-specific activity in individual subjects. So we'll go through this pretty quickly. So with the run structure, we had sort of, these are individual trials. So these were pseudo-randomized between alphabetize and remember, or manipulate and maintain. And those were all sort of in the same run. And then we have a separate run of action versus non-action trials. We basically, all of the results that I'm going to show you are sort of within subject. So what we would do is we would sort of use the functional localizer to get our hotspot, which isn't actually overlaid here, but then we would use actually the nice thing about this bold and vaso thing is that the ratio between the two gives you this nice T1-like contrast. So you can define your layers kind of directly in functional space, and you don't have to try to register a T1 scan to these functional images. And so that really helps sort of preserve, again, the spatial specificity. So all of the layer masks were defined individually for each subject, kind of based directly on this functional contrast. And so what I'll show you is basically we extract kind of average signal values from each of these layers, and that's what we are statistically comparing. So we never sort of like we never try to normalize brains to one another. We do everything in single subject space and just pull out the numbers and then contrast those. And so you'll notice here that we basically split things into 2 layers. It's difficult to sort of drill down and get all 6 cytoarchitectonic layers. Sometimes you can sort of get a middle layer in there, but because we didn't have strong hypotheses about what layer 4 might be doing or what layer 4 is ever doing in a region like this, we just sort of left things as superficial and deeper. So,
and some people even argue that layer 4 is not really something that you can find particularly easily in the frontal cortex, right? Like in the motor cortex, it's supposed to be layer 3B that receives the more sort of core-like thalamic inputs. And so it's sort of like hard to know what it would even mean to like carve it up at that level.
Exactly, yeah. So, and that's actually, I have like a slide on that too, is like the difficulty of sort of normalization to cytoarchitectonic layers, specifically in these higher order regions. So anyway, 2 layers, superficial and deeper. And then for each subject, we extracted a mean time course per layer per condition. So alphabetize versus remember, and then this is what we're doing our stats on. So just to remind you here of our hypotheses, this was what we thought we might see. And this is, I think, the study that I've done, like the biggest, the one study I've done in my career that like the hypothesized activity actually looked like really similar to what we saw. So that was pretty cool for someone that's used to sort of like exploratory analyses of big data sets. But yeah, so basically we saw activity that was more or less totally in line with what we hypothesized. So, and these are contrasts based on the vaso-derived time courses, but basically you can see in blue here, this is activity during the alphabetize or the manipulate trials. And so you can see, again, the TR is kind of slow, so it's like a little bit of a jerky time course, but you can see clearly that manipulation evokes more activity, specifically in superficial layers during, manipulating evokes more activity in superficial layers during the delay period than does simply remembering or maintaining. And then also in the superficial layers, we see no difference between the action and non-action trials, which is as expected because all of these trials were alphabetized or manipulate trials, and they only differed based on whether people had to make an action at the end of the trial. So this is what we should have seen here, and in fact, it is what we saw. And then looking at the deeper layers here, we see a little bit of a rise during the delay period, and then sort of another little bump here on the response, but not a strong difference between maintaining and manipulating in the deeper layers. Whereas the biggest difference in the deeper layers came during the response period, and specifically we saw higher activity when people did actually have to make a response versus not. So essentially our hypotheses were nicely confirmed with these vaso results. We can quantify this a little bit more. So this is just the stats to sort of prove to you that this is all statistically significant. So we see this interaction here in the superficial layers such that alphabetizing evokes more activity than remembering, specifically during the delay period, but not during the response. The superficial layer difference between action and non-action was not significant. And then down here in the deeper layers, we see sort of this slight crossover effect where deeper layers are a little bit more active at the response when you've had to alphabetize rather than simply remember. But the biggest difference in deeper layers comes between action and non-action trials, specifically at the moment of the response.
Emily, do you have a slide where you show where you are exactly in the brain? I'm trying to see, is this dorsal lateral prefrontal cortex, or it looks quite posterior, right? Or, I mean, where are you exactly in the brain?
Yeah, so it's actually really hard to show an average of where we are across everybody, because it's really hard to like normalize these data to some kind of template. So I'll show you in a few slides. I have some single subject results that are a little bit easier to figure out where we are. We are in sort of, I would say we are in the more posterior aspect of DLPFC, but we are not in the frontal eye fields. That was a concern of ours for like a little while doing the study, but we are sort of able to separately localize that. And so I wish I had a better figure for this. Okay. But we're sort of, we're kind of in the posterior aspect of it, like right around, I believe like the border between BA9 and BA46, but the anatomy is actually really variable in individuals too, which is another challenge here. So but it is, I think, what most people would consider DLPFC, which is obviously a big territory, but yeah. So this is just
Can I ask something quickly, Emily, just on the task?
Yeah.
Are there, for the alphabetized version, are there like difficulty levels? Could you imagine there are situations where the items are pretty much alphabetized already and you don't have to do very much work to get them into order, versus ones where you've got to flip 2 or 3 different pairs and switch them around? Such that like, or like even another way to think about it would be there's ones that are close together and ones that are far apart. So like it's really hard to remember some of the combinations, whereas other ones are really intuitive.
Yeah, that's a really good question. And even as we were sort of piloting this task on ourselves, we did sort of notice that, because the letters themselves were actually completely random. And so you could just by chance sometimes like get a group that was by virtue of how they were distributed through the alphabet, easier or harder, or by virtue of their original order, easier or harder. We didn't try to manipulate that at all, mostly because this data, I should also just like say this throughout, this data is like still really, really noisy. And so you need a lot of averages. And so you basically just need like a lot of trials going into this. And I think it would have been, I mean, I think we at 1.2 talked about doing like a parametric load manipulation. So like 4 versus 5 letters or something like that, to see if we could see kind of like parametric increases, which would obviously be an even stronger, nicer test. But at this point, we just, we sort of sacrificed that precision just to sort of be able to get more averages. But I think that's a really, I think it'd be really cool to do that, especially now that the technology has come so far, even since we did this.
Yeah, I was just sort of thinking it would be fun to think about what that complexity challenge would add on top. And you can think about increasing the complexity in a number of ways. Obviously, it makes the task a bit more nuanced, but you could start messing around with the perceptual overlap of the letters, such that they're like sort of smushed together a little bit more versus spread apart. And that could add a bit more of a challenge, or the contrast and noise is a little bit harder. So you've got to like really resolve out whether or not you've actually seen a B, or was it actually, or it's a bit harder with letters because they're a bit more distinct. But if you had digits, let's say you had like a 6 or an 8, they're a little bit harder to tell when the contrast is low. So anyway, I was just sort of thinking that it'd be fun to think about what kinds of challenges that brings for the brain, and how the brain would resolve those kinds of patterns of uncertainty. Maybe you need a broader area of the dorsolateral prefrontal cortex to sort that out, or you need to hold the items longer over a delay and really kind of precisely encode them such that you can, manipulate them and still have a good answer at the end. There's lots of really fascinating questions in that space.
Yeah, totally. I mean, I think that could be a really interesting thing if you could also get, and I think we totally could these days, but, getting visual areas in the same field of view and being able to see some kind of crosstalk, as you're just trying to resolve perceptually what they are, and then also maintain them or manipulate them, and etc. So
it reminds me of the problem faced by a lot of kind of mouse labs at the moment, where they've got these really cool neuropixels probes where they can stick in the silicon kind of it looks like a depth electrode, right? They stick it in, let's say, visual cortex, but if they plan the trajectory just right, they can go through the pulvinar and then down into the superior colliculus or something, and they can get data from all the way across these kind of really interesting axes of the brain. And you've got a similar problem here where you're like, what is the most interesting plane that we could be in such that we get the area we want, but also some other cool areas? Because I think you're right that it's going to be the coordination between those areas that's going to be really, really important to kind of really unpack these mechanisms. Because it's not like the DLPFC sitting in a dish could do this work, right? It needs to be embedded within the rest of the brain and to be coordinated with parietal lobe and probably a bunch of subcortical structures as well. And it's just really hard to do those analyses.
Yeah. At one point, Renzo was sort of reanalyzing these data because in some subjects we did have like our slab sort of hit a decent area of parietal cortex too that also seemed to have a hotspot to this, and we didn't really have quite enough power either subjects wise or like good signal within subjects wise to really like make much of it. But and I think very much these days, like we could have, if we went back to this paradigm, we could probably get pretty decent data across maybe not the entire brain, but like a good chunk of it, and we could get sort of everything in one. So the problem is I just don't have a 17 anymore, but I'm playing the long game. Hopefully that'll change. Right, so this is just sort of more stats about the difference of differences also being significant. I'm gonna skip that unless people really want to see it, in favor of showing you these activation maps at the single subject level. So these are these come from our higher resolution protocol, which was our voxel size here you can see. And so this is data from, again, a single subject, and these are basically the contrast between alphabetizing versus remembering in green, and you can kind of see that tracks along the sort of surface of the cortical ribbon here, and then the signal during responding minus baseline. So this is actually collapsing across all the trials, I believe. It's not specific to the action versus non-action trials, but just basically anytime they're responding versus the intertrial interval. And you can see that that's like pretty nicely localized here, this red stripe deeper to the deeper layers. We can see this in both bold and baso. And then this is the layer mask that we used. Renzo has this sort of crazy massive upsampling and then downsampling technique that he does. But so we can see this pretty clearly in one single subject. We can also see it basically in all of, we scanned only 5 people using this protocol, but we were basically able to find this in everybody that we looked at. But you can also, this kind of gives you an appreciation for just how variable the anatomy is. So it's challenging to sort of localize these things. I think if we hadn't had the online localizer kind of showing us where to look, it might have been harder, to be honest. And that's another challenge, I guess, with like whole brain stuff, is you still probably do want to have for these higher order tasks that aren't quite as like that don't hit you over the head quite as much as like a checkerboard and visual cortex or something like that. It can sometimes be challenging just to localize where you want to be. And so having this is a pretty strong task in the sense that, you know, back in the 90s at 1.5T, they were seeing like 4% signal change in DLPFC, you know, for this particular task, which is one of the reasons that we chose it, is because it is sort of a strong task. So yeah, having that extra bit of functional localization to sort of show us where to look, I think was key here. But once you kind of know where to look, you do see these nice patterns even at the single subject level. So that was, you know, basically that first paper. Our conclusions were, you know, basically that we more or less confirmed our hypotheses, which were heavily based on the monkey work, which was that we saw increased activity in superficial layers, specifically during the delay period. And we believe that that might be due to local recurrent excitation among the layer 3 pyramidal cells specifically, but also to Mac's point, potentially some long range cortical connections, for example, with parietal cortex. And then we saw stronger activity in deeper layers during the response. And, you know, this could be due to a lot of different things, like, for example, motor control, action initiation and monitoring, or some kind of feedback signal like corollary discharge. This is where, you know, sort of we pay the price of only zooming in on this one area in this one study. But at the very least, I guess this was consistent with sort of single unit recordings or, you know, individual aerial recordings from the monkey work. So it was just sort of nice to see that confirmed. And then, yeah, I mean, there's tons of places that we could take this. I initially was interested in this work because there's been a lot of findings sort of at the molecular and genetic level about schizophrenia being, patients with schizophrenia having preferential disruptions specifically to this layer 3 circuitry in prefrontal cortex, and that potentially underlying maybe not only some of the cognitive symptoms, but also some of the more affective symptoms as well. So it would be awesome to do this in patients. I think the 7T environment is still a little bit challenging on that front, but it would be very, very interesting to see if, so people have observed weaker activation during working memory and DLPFC in patients with schizophrenia, but it would be really cool if we could localize that specifically to these, what we think are these layer 3 pyramidal cells, and kind of tease out the layer specificity of those deficits. So stay tuned. But so that sort of was our first foray into this. And then I figured I would sort of throw in some of the more recent work to give you guys a flavor for the true state of the art, since again, the field is moving so quickly. So even in the last couple of years, there's been a lot of changes. I think there's still a lot of challenges of doing layer-specific fMRI and association cortex. This is one we already sort of touched on, but one of the biggest challenges is that you really need an area-specific normalization of what we call layers in fMRI with the ground truth cytoarchitecture. So this is a figure that Brenzo largely made. And you can kind of see, so these are all sort of different ROIs selected from association regions. You can see the sort of larger map on the left and then a zoomed in version. And then these profiles here, basically what you're looking at from left to right is increasing cortical depth. So the layer in pink here roughly corresponds to layer 1. In teal are layers 2 and 3, layer 4 to the extent that we can identify it in these regions in pink, and then layers 5 and 6 in green. And there's sort of a couple ways you can try to map between our fMRI voxels and more specific layers. And well, I guess, so the first point is you can sort of see that these layers take up different proportions of the whole depth in different regions. So, and not only that, but the signal itself can be functionally dominated by different layers in different regions. So for example, this region here, Broadband area 40, the trace in red here is basically the correlation between a layer specific signal with the area wide signal. So you can sort of think of it as like how much layers contribute to the overall signal in that region. And so some regions seem to be like strongly dominated by deeper layers, like this one here. This one has sort of this bimodal profile where you see basically strong activity in both superficial and deeper layers, but not so much in layer 4. This one seems to actually be more, in theory, feed forward dominated. You sort of see this peak here around what we think might be layer 4. But yeah, you can just sort of appreciate the heterogeneity, not only in where the layers actually are with respect to, you know, our voxel grid and MRI, but also how much each area could be dominated by theoretically feed forward or feedback signals, or at least sort of more layer specific signals. So it's a lot of work basically to normalize these things so that you can more correctly interpret your fMRI findings based on kind of ground truth cytoarchitecture.
Are they pretty consistent across subjects, those little red waveforms? Like, can you say something like, in general, BA6 is, you know, a granular dominated region versus BA44 is infragranular, and then kind of carry that forward? Or do you think there's going to be a lot of individual differences in those kind of patterns?
Yeah, that's a really good question. I mean, I think there's sort of two levels to that. It's like, first we have to assume that we can actually identify the same region, right? So like there's differences in anatomy, and there's also differences in functional anatomy. So like where, you know, certain functional specialization profiles map on, even if two subjects have the exact same sort of gyral profile, you know, where you are along that gyrus could be more sensitive to, you know, a certain function than, or yeah, that could be like a slightly different mapping across people. So, I don't know.
It's really hard.
It's really hard. Like my sense is like, there should be, I mean, you would hope that there's some consistency across people, but to the extent that you see variability, it could be quite interesting, but it also could be really hard to convince yourself that, you know, you're actually in exactly the same region in two different subjects, and therefore that like the differences that you see are, you know, reflective of some sort of true functional difference, as opposed to just like being in a slightly different place on the map. You know what I mean? Yeah. I mean, I guess you could do sort of larger averages over some of these areas and see if you could get like general differences there. But yeah, I don't know.
It's also pretty easy to imagine this having a real state dependence too. Like it could be that the bold, when you're at rest in a association region, has a particular flavor of it relates, let's say, to, you know, granular regions. But then as soon as you start to like push the system and the super granular regions become more important for the context you're working in, now all of a sudden the whole bold looks more like that subset of groups or something like that. And so the complexity here could actually really carry the day and be very hard to refine down to these kind of like heuristic mappings that we'd like. But yeah, brain's complicated.
Totally. Yeah, totally. Yeah, I sort of already said all of this, but there's just so much inter-individual variability. I mean, it's true all over the brain, but it's especially true in these higher order regions. So it becomes pretty challenging to look at. I mean, you can look at individual differences, which are sort of my other bent, but it's really hard to sort of do it in a way that's rigorous and that you convince yourself that the variability, that you're actually attributing variability to like the right sources, or that you're able to interpret it rigorously. So, but if your goal is just to do a study at the group level and not look at individual variability, I think sort of having these functional localizers has been really helpful for these more cognitive-y areas that you can't identify based on known anatomical landmarks. And then, yeah, so I guess this also sort of relates to Mac's point about, you know, beefing up the paradigm or making the paradigm more interesting. I think it's challenging. I mean, it's certainly where we want to get to. I think right now you just need so many more averages than you need even at like 3T, for example, that you end up kind of being limited. You're really still limited by SNR here. And even though the acquisition, the brain coverage and other features of the acquisition continue to improve, it's still a pretty signal-starved world that we're playing in. And so you just need a lot of averages. The TRs are still pretty slow, especially if you want to do this sort of bold vaso thing. And so it becomes harder to do sort of more nuanced event-related work. We sort of artificially stretch these trials out even more than maybe we would have if this were a more standard 3T study in order to sort of get enough time points at each period of the task. But you can imagine that might start to feel a little bit unnatural, or sort of like, depending on the cognitive task that you're interested in running, it might be more or less amenable to that sort of temporal stretching that you might want to do if you're sort of playing in this space. Yeah, but that brings me to the place where my two worlds sort of start to overlap more, which is you could also just use movies. So I wanted to show you guys this, which again, this is all Renzo. We collaborated loosely on this, but it was mostly him. But he started doing some of the stuff before he left NIH, which was really fun. So he was having people watch movies during a pretty sizable, it wasn't whole brain, but it was like pretty close at that point already. And he took movies from the Human Connectome project. So they have this movie acquisition also at 7T, although it's not layer specific. And so he basically had a reference time course that was derived from 93 independent subjects. And this was a 15-minute movie clip that people watched in the scanner. And it was comprised of individual like 3 to 4 minute clips within those 15 minutes. So there was sort of varied content and whatnot. And he basically used the reference time courses from the Human Connectome participants to, he was actually trying to get at individual differences here. So he had, I can't remember how many subjects went into this on the layer specific side. I think it was maybe around 10 or so. But he ended up sort of clustering I think it was actually more like 15 or 20, because he had a decent number in each of these clusters. But yeah, so essentially what he was able to do was take these reference time courses from this interparietal sulcus area here from the Human Connectome project participants and use them as regressors for the layer specific data. And he was able to identify sort of 3 clusters of people based on layer specific activity signatures in response to this regressor. And the interesting thing he found was that, largely, everyone's getting the same perceptual input. So you see sort of mostly they're pretty well synchronized. The frame where they were, where there was the most, or the biggest differences across these 3 clusters of people was this frame here, where there's like a kiss. So it's sort of like an evocative frame, I guess. This was an exploratory analysis, I should say, just kind of for fun. But I think the most interesting thing here is that these 3 clusters of people, so in black, gray, and blue, most of the differences, at least in this black cluster, were in the superficial layers. So this graph here is moving from left to right on the x-axis. You're going from superficial to deeper layers. And this is sort of the signal change in response, I think, specifically to this frame. And you can see that you have sort of the most individual differences arising in these superficial layers, and it's more stable down below. So again, exploratory, but we're still kind of piecing through this. And I think the movie stuff is nice because you can actually get multiple averages. It's a little bit confounded because watching the same movie twice is not going to be exactly the same experience, but you can sort of have multiple acquisitions and then be able to, because you have that precise time locked stimulus, you can, it makes sense to just do a straight average across acquisitions, and that can help boost signal in a lot of ways. This is another really cool application that Renzo's been working on. So this idea of sort of, this is sort of the promise of layer fMRI in a lot of ways. So being able to map feed forward versus feedback driven activity. And so here what he's doing is this sort of hierarchy mapping by means of a seed-based layer-dependent clustering analysis. So what he's doing, basically this is visual cortex in a single subject, and he's sort of iteratively placing a seed in different places And then he has these sort of templates. So basically the idea is if a layer, or if a region or a column, I should say, if a column is strongly dominated by feed forward activity from that seed region, you should see stronger connectivity with the seed, specifically in layer 4. Whereas if a column is driven more by feedback connections from the seed area, you should see sort of stronger connectivity in both superficial and deep layers, but weaker connectivity in the middle here in the granular layer. So he has sort of these templates, and so he's stepping this seed essentially along the visual pathway. So in this first panel, the seed is in thalamus. It's out of the field of view. You can't see it, but you can see basically everything in V1 has this nice sort of feed forward match. So it's the layer that's most strongly correlated with the thalamus in all of these areas is layer 4, as you might expect. And then you kind of step the seed along. So here now it's right here in green, and you can see everything sort of up the visual hierarchy, so to speak, is still feed forward dominated. And these, like, I guess, very early areas in V1 are now feedback dominated by that seed. And so hopefully you can kind of extrapolate this out, but basically then you step it a little bit further and you see that you have more feedback dominated activity here in the middle. And then as you sort of move out I guess this is more or less the ventral stream. I don't know exactly what to call it, but as you sort of move up into higher order association visual regions, you start to see more feed forward dominated activity from that seed. And then you sort of step all the way through. And this is the end where you're all the way out here in sort of lateral occipital cortex. And you can see that basically everything behind that, so to speak, in the visual hierarchy is feedback dominated from that area. So I think this is
cool. Awesome. Love it.
Yeah.
Do you know how much wiggle room you have with the thalamic seed? Because if you moved into an area of the thalamus, let's say you were moving out of LGN into an area that has more of the diffuse matrix-like projections, you might predict that you'd get the feedback pattern more than you'd get the feed forward pattern. And you could kind of map out the thalamus, or at least the kind of posterior thalamus as it interacts with the field of view you have there in a really neat way. Maybe I can ask Lorenzo, but that would be super cool.
Yeah, that's a great question. That's a great question, and he would have a better answer than I do. But yeah, I think this is like really promising, and this is kind of where we, just want to be a little mindful of time. Of course, I'm spending longer on this than I planned to. I think this is my last slide on this. So, and then finally, you can start to look at layer-specific functional connectivity sort of across a wider spectrum of regions. So the slide before was just sort of seed-based and moving the seed around, and now he's kind of looking at these more like macro level connections between larger cortical regions. And he's basically building these layer-specific connectivity maps. So now you sort of have an extra dimension to your matrix where you have not just like a region by region matrix, but you have layers within each region. And so you can kind of look at whether connectivity between two regions is driven more by certain layers than others in one region versus the other. So to make that a little bit more concrete, he's showing some interesting examples here. So this is a sort of visual network between V1 and V5. And so you can see kind of within each region, there's a lot of connectivity along the diagonal. So layers are connected within each other and then also connected to other layers in that same region. But when you look across V1 and V5, you're seeing stronger connectivity between superficial V1 and deeper V5. So he's sort of interpreting this to mean that V1 is getting feedback in the superficial layers from V5, while V5 is getting this feed forward input more into the middle and the deeper layers, which is, I guess, the direction that you might expect. And then another example from the sensory motor network showing that, so BA4 is a motor area, and then BA3B and BA1 are sensory areas. And you basically see this strong signature such that BA4 is only really connected to or getting input, so to speak, from BA3 and BA1 in the superficial layers. So this stripy pattern here. And that's consistent, I believe, with what we know from track tracing and I guess more specifically from monkey studies, I suppose. And then here in the everybody's favorite default mode network, this is kind of an interesting one too. So this is looking at medial PFC, PCC, and interparietal cortex. And I guess the most notable finding here is that PCC actually seems to be strongly middle layer dominated, whereas other layers are seemingly more feedback driven. And I think the suggestion here was that, I mean, so PCC has been identified as a hub in sort of any number of ways. So it could be that the sort of input to the DMN comes into PCC and then it sort of spreads forward into these other regions. I mean, again, these are still pretty exploratory analyses, but I think this is kind of like the promise of what this technique might be able to tell us, is like actually starting to be able to infer more directional connectivity, which is really exciting. And then finally, the frontoparietal network, there's actually less, I mean, so these were defined, I should also say, based on ICA components. So there definitely is, you know, already some coherent fluctuations between all of these regions that are being examined. In this network, there was actually less crosstalk between this frontal and parietal region than you might expect. So they were sort of strongly connected within regions, so frontal with frontal and parietal with parietal. And there wasn't as strong of a layer signature across these two regions as we saw in these other networks. So anyway, but yeah, I mean, there's tons of open questions about this. You know, how state dependent is this? This was all based on resting state, so can we modulate this with a task? But I think the point of this is just showing that like this type of thing is now becoming possible, which I think is really exciting. And so yeah, there's like a ton of places that we could go with this, but this is more or less the state of the art, courtesy of Renzo. And he's a great speaker, so if you guys want to learn more about this, I would strongly encourage you to invite him as well.
Awesome, thanks, Emily. Should we maybe just quickly pause here and see if anyone has any questions they'd like to ask? Is this a good stopping point just for that, Emily? Or
yeah.
Yeah, great. Does anyone have anything they want to jump in or ask about? I mean, this seems super cool. We've been talking a lot in the lab about how much we want to understand better what you can do in sort of layer fMRI context, because a lot of the modeling work that we are trying to do, and also a lot of the kind of source literature that you go to try to inform those models, they have this kind of cell specificity, or even like maybe even less a layer specificity to their recording. So they'll say something like, okay, in layer 2-3 cells in the mouse, in this context, all this stuff happens. And then we'll go and read about that cell type, and it turns out it gets projections from some subcortical structure or something. So you really, really want to be able to ask the questions of bold data in that, at that kind of resolution. But then when you go out to kind of whole brain bold, you kind of realize you've kind of almost sort of gone too quickly. You've zoomed out so fast that you've lost track of the details. And I think things like these kinds of recordings will mean that we can start kind of inching those fields closer and closer together. So you can say, all right, I reckon in this context, the thalamus was doing this and not that. Go do the recording. Actually, it turns out you're totally wrong. It was doing this other thing, and we can kind of iterate and learn through that. So I'm really excited that this is kind of pushing forward. It's awesome.
Yeah, I agree. Do you guys have a 7T at Sydney?
They're promising to get one. There's a higher Tesla scanner for preclinical at the moment. That's pretty cool, but nothing for humans that we get to play with. They have one up in Queensland, and I think down in Melbourne as well. But
yes, one in Melbourne, one in Queensland. Yeah.
Yeah, sorry. Yeah, Jim would know way more than I would about this stuff, but it's supposed to be coming, right? That's the story. I mean, it's always the story, but
Fernando
was talking about it.
Proposing.
Yeah, it's hard to set them up, and you obviously need a lot of oversight, and you know, you have to take a lot of care to make sure you get good signals out of the scanner. So you know, it'll be a while before we're doing this kind of stuff ourselves.
Yeah, I mean, I can't do this stuff myself either, to be honest with you. I think this still requires like pretty strong physics support or physics collaboration. I mean, I am heartened by, I was actually just on a meeting with Renzo this morning. And one of the things he's really been, so he's back in the Netherlands now, but he's really been working on actually pushing this to work at 3T as well, which would be amazing.
Oh, cool.
So he's been spending like a decent chunk of his time on that. And every time I see him, I'm like, hey, like what's the status? Because it would be, and he knows it too. I mean, you know, there's a decent number of 7Ts out there, but it would just be so awesome if we could get something like this at 3T. Even if you do sacrifice a little bit of the whole brain coverage, like just being able to get even just like a few areas that you're interested in. And yeah, so he's seeing some promising stuff there. And I think it would sort of be a little bit more plug and play at 3T. I think there would still probably be, you know, things you had to work out and things you had to fiddle with a little bit more than a standard 3T resolution study. But yeah, I'm hopeful that those of us that don't have 7Ts yet might be able to still do some of this work in the not distant future. So yeah.
All right, well, in the interest of time, there's nothing in the chat box. So do you want to keep rolling forward with your slides, or if you want to just, we can have a general chat about the layer stuff and leave the naturalistic for another time. It's totally up to you.
May I ask a question?
Oh yeah,
Yeah,
sorry,
please.
go ahead.
Sorry. I'm really interested in the denoising of this type of amazing data. Do we have less noise than the traditional 3T MRI with this 70 setting, or we have more noise? And what kind of denoising steps are involved? Like I can imagine that the previous spatial smoothing would not work on this layer-wise fMRI, right?
Yeah, it's a really good question. So generally, even though the base field strength is higher, you have much less signaling in each voxel, just because your signal is directly proportional to your voxel size by like a factor of 3. So you increase, I think, yeah, Renzo has a good like stat on this, but I think it's like you basically end up with like 1/64th of the signal because it's like a cubic thing. So even though you are like aligning more protons in theory because the field strength is higher, you're just, when you push down to these small voxels, each voxel is just so much noisier. So head motion is a much bigger problem. So a lot of what we were doing at NIH, or like Renzo sort of had this like cluster of like professional subjects almost, who were like the people that he knew wouldn't move very much, because motion is an even bigger deal at 7T, or like any sort of like field disturbance, physiological artifact and stuff like that. So so spatial smoothing is kind of a no-go, although he has this sort of interesting, I sort of alluded to this and I'm probably going to butcher it if I try to fully explain it, but basically what he does is he like strongly upsamples along the
Layer.
axis perpendicular to the cortical surface, and then he does a little bit of smoothing in that space, and then he kind of puts it back into our like more standard voxel grid, and that seems to help with a lot of things.
It's kind of the spatial smoothing along the geodesic surface, right?
Yeah, right. Sorry. So like within layer, never across layer. So he upsamples, he has sort of this like 20 layer, so only within that, only like along the curvature and never across the normal there, so to speak. But yeah, and that's also honestly an area that's constantly moving and changing, and it's also one of those things where there aren't, I guess, that many labs doing this yet, but there are as many pipelines as there are labs. So there's not like similar, I guess, to like other, even at 3T, we have this issue where, everyone sort of has their own flavor of pre-processing. But yeah, I mean, the main challenge is just that you're starved for a signal in so many ways. So you want to be really careful to preserve the signal that you have. And so the less you can do steps that call for like interpolation or anything. So that's why we also we sort of tend to do, or like Renzo's pipeline is to kind of do everything in single subject space and avoid any kind of normalization to a template. Other people, do normalization, and if you have a structural image, it's sort of acquired in the same slice prescription in the same plane as the functional stuff. You can do that to some extent, but yeah, it's a challenge.
Another question is that why do you put the boat and the vessel so just in one sequence? Why don't you separate them? You have more trials. So in the first part, half of the trials you do the boat, and in the second half you do the vessel. Is it possible?
So you could, but you would sort of be losing the main advantage, which is that so in the pre-processing, what Renzo does is he actually uses now I can't remember which direction it goes in, but he's basically able to use one contrast to sort of baseline correct the other. And so having them be near simultaneous is actually an advantage, because he's like sort of processing every frame with respect to, the nearest contrast, or rather I think it's the average across the two, the one right before and the one after. So he is actually able to combine the information in a way that gets us better signal than either would on its own.
Yeah,
And so having that within a trial actually helps you like denoise that particular trial, as opposed to just sort of extracting average time courses from one contrast and then average time course from the other contrast.
Yeah, but the downside is that you have make the TR longer.
It does make the TR longer, which is a disadvantage, but I think the benefit of having them be near simultaneous is that you can sort of do this correction that cleans up the time course in a way that you wouldn't be able to do. I can send you, I'm sort of, he has like a whole paper on this actually, and like the mechanism of this that I should probably share with you rather than explain. But yeah, there is a reason for it, although it does inflate the TRs, which can be challenging for some of these more like faster cognitive-y processes that we might be interested in. So yeah.
Anyway, very exciting. Yeah, very exciting work. Yeah.
Thanks. Yeah. Okay, well, we have a little bit of time. So let me go put on my other hat, which is individual differences and naturalistic tasks most recently. So can you guys see that okay? Awesome. I put your date on here rather than my date, so giving a talk in the future. I'm
so
sure
yeah, that actually, why is South down, right? In all the maps, South is on the bottom. I think South should be on the top. There's nothing.
You guys are ahead, man.
You guys are
front. I mean, it's still July 1st here, you know what I mean? It's like back to the future here. Anyway, so I cut out some background, this for reasons of time. Hopefully I still won't take too long, but if something isn't clear, just stop me. So basically, we've known for a little while now that individual functional connectomes are stable and unique. And so what this looked like in our hands the first time we noticed this result was if you sort of take a whole brain parcellation and you calculate a functional connectivity matrix for a single subject, which is just the correlation between all possible pairs of nodes in a parcellation. And you do this in a large group of people that are scanned under different conditions across different days. And then if you ask the simple question, can we sort of match an individual based on their connectivity matrix acquired in one state to another, can we do that with any kind of accuracy? And it turned out that we could, and that was exciting. And you know, this was well above chance. And then depending on the sort of specific pair of scans that we used, we would get differences in accuracy here. But this is, again, data from Human Connectome Project, 126 healthy people. And so this was, at the time, you know, we thought a pretty exciting and striking result in the sense that it showed us that there was a lot of uniqueness within subjects, uniqueness across subjects and stability within subjects of these sort of whole brain functional connectivity profiles, such that we could identify people regardless of what they were doing while being scanned. There's been some other really nice work from many other groups finding the same thing. I'm just highlighting one example here from Katerina Greton et al. Using data from the Midnight Scan Club, where they also sort of showed a broadly similar thing, so much more highly sampled individuals. But basically they showed that once you account for the sort of stable group blueprint of what functional connectivity generally looks like, most of the rest of the variance in the connectivity matrix has to do with the individual themselves and not necessarily the task that they're performing or just kind of general individual session related variance. Interestingly, they did find that about 20% of the variance, I believe, on average, was due to the interaction between individual and task. So in other words, basically, there weren't necessarily common task signatures across individuals, but there were common signatures of individuals doing specific tasks. And that did account for not as much variance as the mean or the individual, but it did account for, you know, a decent chunk of the variance there as well. And so, you know, there's been a lot of interest and a lot of like really nice follow-ups on this type of work over the last few years. But I think one of the things that's sort of been happening, and there's also been an increased focus, I think rightfully so, on reliability in psychology and neuroscience more generally, and sort of how stable and how reliable are the signals that we measure with fMRI. And all of that is well placed, although I think in our case, you know, we were interested in these individual differences, not just sort of for their own sake, but for what they can tell us about people's behavior outside the scanner. So, you know, could these eventually be used as biomarkers, for example, in real world contexts? And so I think the fact that sort of these two lines of work, so this, you know, these findings of really strong, reliable individual signals, and also some parallel work from our group and others showing that you could use individual patterns of functional connectivity to predict cognitive traits, for example, things like fluid intelligence, sustained attention, even some symptoms of psychiatric illness. These two lines of work are kind of proceeding in tandem. And in a lot of cases, I think it led people to sort of assume that by increasing reliability or by increasing the potential for sort of individual identification, you were also increasing sensitivity to meaningful individual differences that might relate to behavior. And I don't necessarily think that that's the case. And so this is sort of my spiel about why I think that's true. But I think, yeah, this is a point that's like gotten conflated a lot, and I've been thinking a lot about it. So, you know, and maybe this seems obvious to a lot of people, but there's sort of some theoretical evidence that simply by increasing reliability sort of from brain at time 1 to brain at time 2, you're not necessarily increasing sensitivity to behavior or your ability to predict behavior either within or across people. So, you know, and this was a question that we got asked when we first started to do this work, and we started showing the sort of fingerprinting results is like, okay, well, we can identify people, but like, what does that actually mean? You know, is there anything meaningful within that connectivity matrix that would allow us to predict something about that person? And so that doesn't necessarily follow. So you could have a situation where connectomes are unique but not meaningful. And in that sense, we could analogize them to barcodes, for example, where barcodes uniquely identify an item, but there's nothing in the pattern of thick or thin stripes in a barcode that actually meaningfully relates to the item that it identifies, right? So, and then we could also have a theoretical scenario where connectomes are meaningful but not unique. So you could have, for example, pairs or even groups of subjects where they might have very similar connectomes to the extent that we might even mistake them for one another, for example, in identification experiments. But if we were to look at some dimensions of behavior in these people, it could be the case that, you know, these pairs of people with very similar connectomes are also very similar on some sort of behavioral metric. And so in this case, it could be true that even though the connectomes are not unique, they're actually more meaningful in the sense that knowing something about someone's connectome, you know, we could build a model based on people with similar connectomes to predict something about their behavior. So that's just sort of a theoretical argument as to why these two things are, I think, pretty clearly do not entail one another in my mind. And then there's also a lot of empirical evidence now that reliability does not, or increasing reliability does not entail an increase in behavior prediction, in the sense that if you just look at the individual edges or connections, this is the whole brain matrix. This is work by Stephanie Noble and co-authors from a few years ago now, but basically they just sort of directly tested this hypothesis. So what they're showing here is the correlation between an edge's reliability, so sort of test retest reliability, versus its relevance for behavior prediction. I believe this was fluid intelligence. And you can see there's like basically no correlation between how reliable a single connection is and how important it is for behavior prediction. So this suggests that if we were to optimize for reliability, we might not be optimizing for behavior prediction. And so I'm gonna, I guess, walk through a little bit more empirical work now that that I've been doing on this. And so this is work from a couple of years ago now. But you know, I think one of the messages that it would have been easy to take from the fingerprinting study is, well, it doesn't really matter. You know, we can identify people regardless of what they're doing while being scanned. So, you know, who cares what state we have them in while they're in the scanner? But it turns out that brain state actually matters a fair amount. And so when we drilled down into this, this is now sort of a replication and extension of the identification or the fingerprinting findings, where what you're seeing here is basically our ability to match subjects across different pairs of brain states or scan conditions. So matching from a target to a database, these were scans acquired on 2 different days. There's 2 rest scans denoted by R1 and R2, and then there's 7 different cognitive tasks that you can see here. And so the main takeaway, at least for me from this matrix when I first saw it, was there's a lot of variability here, right? So, you know, there's pairs of scans that were really well able to match people, and then there's also pairs that, you know, were not very well able to do that at all. And one of the striking things here is that if we look across the 2 resting scans, so rest 1 to rest 2, we do pretty well. We get people with about 70% accuracy. And I should mention here actually that we've truncated all of the data in this analysis to the shortest task. So this is actually based on very little data. It's only based on about 4 minutes of data per subject. And chance levels are really, really low. So all of these identifications are significant, but there's obviously still a lot of variability in our identification rate. And so rest 1 to rest 2 does pretty well, but it actually doesn't do as well as pairings of certain cognitive tasks. So for example, if we try to match a person's working memory scan to their scan during this relational matching task, which was sort of one of the more demanding cognitive tasks that they included in the Human Connectome project, we're able to do that with 83% accuracy. So, you know, you only get single numbers from these. It's hard to put statistics on this, but you can sort of start to see that, you know, there is, so even though rest in theory is the same state both times, although we know that there's a lot of variability in terms of what's going on at rest, even though that's in theory the same state, we're not getting as good accuracy as we are between 2 supposedly different task states here. To quantify this a little bit more, this is actually kind of surprising to me, at least when we first ran it. So this is a plot of all of these 9 scan conditions here. And on the x-axis is basically overall how similar people look to one another. So if I just take everybody's connectivity matrix and correlate it with one another in that state, how similar overall are the matrices from that state. And that's plotted here against what we're calling the mean database identification rate. And that's literally just the average across each of these rows of this identification matrix. So on average, how well are we able to identify a person in that condition when paired with all other conditions as possible targets? And to cut to the chase, what you see here is a strong positive relationship, and that's implying that tasks or brain states that make subjects look more similar to one another actually make better databases for identification. And that was surprising, at least at first to us, because you might think that you would get the best traction on individual differences by putting people into a state that kind of pulls them apart, right? That separates them maximally and makes them look really distinct from one another. But it turns out that the states in which that's true, which happen to be the 2 resting runs as well as this motor, which was a really simple finger tapping task, you're making people look less similar to one another, but you're also in some way making them look less similar to themselves, at least less similar to themselves in these other sort of more structured task conditions. And so this was just sort of, you know, an interesting empirical thing that we noticed here, such that, and I guess the way that we were interpreting this and still are interpreting this to some degree is that tasks overall, and, you know, we're limited to inferences about the specific tasks that were run in this study and all of that. So, you know, there's many other brain states that we could think about, but at least within this group of task conditions, it seems like task is sort of acting as a lens almost through which we're projecting people, and we're sort of constraining the overall variance in a way that makes the remaining variance across subjects more meaningful and more stable and more trait-like. So by sort of having a task on board, just sort of any kind of task, you're putting people into this more sort of stable state where the functional architecture is more similar to themselves doing another task than it is to these more unconstrained states. And whether that's desirable or not desirable, I think that depends on your scientific goals, but I think this was a pretty striking result just from an empirical standpoint of, you know, if your goal is to maximize sort of reliability and stability, you know, rest may not be what you want to be doing. and other people have sort of converged on this in different ways. But to sort of tie it back to the larger point I was making before, you know, I don't see identifiability as sort of an end in and of itself. I see it, you know, I think the real value in studying these individual differences is in their ability to predict behavior. And so to to that end, we're also seeing that task states do a much better job at predicting behavior than rest does. So this is the same data that I was showing on the previous slide from HCP. And here, basically, what we're doing is we're training a predictive model to take in functional connectivity information and predict different cognitive traits at the single subject level. So we're predicting fluid intelligence, verbal IQ, and a working memory measure that was done outside the scanner. So these are all behavioral measurements taken outside the scanner, and we're trying to see if we can predict them based on functional connectivity from these different brain states. And all of these, I should say, are statistically significant again, but you can see that the 2 resting runs actually do the worst across the board at predicting these cognitive variables. And the ones that do the best in this data set happen to be working memory and language and gambling. But I mean, this is pretty striking. Like even a simple finger tapping task, you're getting a better prediction of fluid intelligence than you are just from rest. And so there's a lot of, questions about if, whether this is due to just some sort of low level arousal effect, like are people just sleeping at rest, or that, like where exactly is this additional predictive power coming from? But, I think, it's a pretty striking result, and it's one that's been replicated by other groups in another data set. So this is another demonstration of this from Abby Green and Todd Constable's group, where she's sort of replicating these findings on the HCP and showing that this is variance explained also for a fluid intelligence measure, and connectivity during, in this case, the gambling, working memory, and emotional tasks in this subsample explain the most variance in fluid intelligence. Resting state was down here doing pretty poorly, all things considered. She saw the same thing in a developmental sample from the Penn neurodevelopmental cohort. So these are children and adolescents. There were only 3 conditions available, but there was a fluid intelligence measure. And she also sees that the working memory, that the task scans essentially do a lot better than the rest scan for predicting fluid intelligence in particular.
So Emily, do you use, what features do you use for the prediction, like all the features in the correlation matrix, or a subset of them?
This is using our what we call our connectome-based predictive modeling, our CPM approach. So it's a data-driven feature selection. So we throw all of the edges in, and then we basically have a feature selection threshold. That's kind of a hyperparameter that we can vary. And then the final input to the model is just basically an average across all of the correlations that are positively, all of the connections that are positively correlated with the behavior of interest in the training set. So we separate out the positive and inverse correlations between the edges and the behavior, and then we combine those. Well, we either train separate models or we combine those into one model, but it basically ends up being a simple linear regression at the end of the day. But it's, the feature selection is all done in a data-driven way. So you do, one caveat with this is you can get different sets of edges that are sort of doing the work of prediction across the different folds and stuff like that. And so there's additional questions we could ask about, how stable those features are, and if we wanted to actually go back and interpret them in an anatomical context, which we tend to try to do. It's hard. They end up being pretty messy and all over the place. But it is, the results I'm showing here, they are based on a whole brain sort of data-driven model. Yeah, so the model has everything available to it, and it can select different things based on, what happens to be correlated in the training set, but we're not like artificially restricting that at all. And it's the same across all the conditions. Okay, so, right, so this was like a couple years ago now, and at this point I started to get really interested, like many people before me, and also now, in these naturalistic conditions. So things like having people watch a movie or listen to a story in the scanner. And we thought this would be a really interesting challenge specifically to the identification experiments, because dating back to sort of the seminal work of Ori Hassan and many other people that have been working with these stimuli, one of the dominant ways these stimuli are used is to kind of synchronize everybody, right? So one of the really interesting things you see when you have everybody watch the same movie, you see this really strong synchrony in many regions across people and the activity that's evoked to that movie. And so we thought this would be an interesting challenge to see, is the individual signal still there while people are watching movies, which, you know, might be the state where they are looking most similar to one another, at least in some parts of the brain. So we had a data set that we were able to test this on in collaboration with Tammy Vanderwall at Yale at the time. And she had, this was a smaller data set than HCP, but she had 34 healthy adults come in for 2 sessions where they did a resting state run, this run where they were watching a really abstract visual movie, and then a clip from Ocean's 11. And so you can kind of see, this clip did have sound. I think it's not playing right now, but these were two very different movies. But it turned out that if we calculate functional connectivity profiles for each subject in each one of these runs in each session, and we try our sort of simple identification algorithm on this, we're able to match people with quite high accuracy across these different conditions. So not only between the same states of sort of rest 1 to rest 2, the first viewing of inscapes to the second viewing of inscapes, the first viewing of oceans to the second viewing of oceans. So along the diagonal, sort of within the same state, and we're able to do that quite well. But even off diagonal, so even going from rest to a movie or between 2 different movies, it seems like the identifiability in the connectivity matrix is preserved, or, you know, even in some cases maybe enhanced. Again, you kind of only tend to get one number from these, but you can see at least numerically the identification accuracy is actually highest for this Ocean's 11 clip, which also, you know, per prior work would be the one that we expect to evoke the most synchrony across people, since it's sort of the most rich, dynamic, engaging stimulus. So this was encouraging for me, since I was hoping to use these stimuli to sort of study individual differences and tease out some behaviorally relevant variability that we might care about. So this is the last set of results that I'll show you, I promise, but this is sort of the most recent place that we've gotten to with this. And so we wanted to know, I guess, you know, going back to sort of the grounding question, at least for me, is not just like, okay, are these individual differences there, but do they actually mean something? And so can we use them to predict behavior? And so turning again to the Human Connectome project, this time the 7T data set that does have movie watching, as I alluded to before. This is a smaller sample than the full HCP, but still a decent number of people, depending on who you ask these days, but enough to start to test some hypotheses. So about 176 healthy young adult data went into this. And because the HCP, they have a ton of different phenotypic measures, we kind of narrowed down into 2 domains. So we wanted to see if we could predict broadly cognitive ability and also socioaffective traits. And so these were 2 different measures that we constructed, basically just doing a big PCA on all of the measures in these domains. So cognitive ability had things like fluid intelligence, working memory, vocabulary, processing speed. It was sort of a big amalgamation of a lot of different cognitive tasks. So we're not able to get super granular, but we also didn't want to run like 100 different models. So this was sort of a sort of general factor that we extracted here. And then we also had a general factor that we extracted from the sort of socio-affective traits, which were all self-report scales. And in this case, we're mostly based on the NIH Emotion Toolbox. So the structure of the data set, people came in for 2 days of scanning. They had a 15-minute rest run, which was always done first. And then they had these 4 different movie runs that each lasted about 15 minutes. And so I mentioned this when I was talking about the layer-specific results, but the key thing to know about each of these movie runs is that they're each comprised of a series of 3 to 4 minute clips. So there's like 4 to 5 clips within each of these movies, and they're separated by 20 seconds of rest. So basically our goal here was just to empirically test whether movie watching would give us better, or I should say functional connectivity data acquired during movie watching would give us a better prediction of these 2 behaviors than functional connectivity during rest. And that's more or less what we saw to be true. So here what you're seeing is the prediction accuracy measured as the correlation between observed and predicted behavior. The dots in purple come from movie watching runs. These are 100 iterations of the model using different train test splits, and they're all compared to a null distribution here in gray. And then the dark gray models come from the resting state runs. So and this is session 1, so rest 1, movie 2, and this is session 2. And so these predictions are for cognitive ability, and you can see that movies do give us a boost, in some cases a pretty dramatic one, in prediction accuracy over rest. Overall, the data from session 2 was a little bit worse for some reason. I think there may have been more data quality issues on that second day. I'm not totally sure. But we actually couldn't get significant prediction from the rest run on in session 2 at all. And we did see some accuracy with movies. So that was kind of interesting. And then extending this to the socioaffective tendencies, these tend to be harder to predict overall. I think we've seen this in our hands. I think other groups have seen this too. Again, I'll remind you that these are self-reports as opposed to behavioral measures. And so, you know, it just tends to be, I guess, harder to predict these things in many cases. And so the only run that we were able to get a significant prediction of socioaffective tendencies was movie 1 in this first session. And actually none of the runs in session 2 gave us a good prediction of socioaffective tendencies. But this was sort of some initial confirmation for our hypothesis. We did some checks to try to rule out sort of general confounds of low. So one obvious question is sort of like, are people just asleep during rest, or like, are they moving more? All of these things. We did a bunch of stuff to try to look at that. We're pretty convinced that it's not a sort of global arousal factor. I'm going to skip through this for reasons of time, but if people have questions, I'm happy to come back to it. Because I think the more interesting thing is that because these runs were comprised of these individual clips, we can separate out and calculate connectivity on a clip-specific basis, and then train our models just based on activity from these individual 3 to 4 minute clips and see how well we can predict, in this case, cognitive ability from individual clips. And so you can see there is actually a decent amount of variability in how the clips are doing. These later runs, again, was much more challenging, but some of these clips, specifically in the second movie run, which came from the Social Network and Ocean's 11, actually did some of the best prediction for us. And if we extend this to the emotion or the socioaffective trait measures, again, overall, much harder to predict these, but some individual clips are actually giving us pretty good prediction. So again, this clip from Social Network and this clip from Ocean's 11 did quite well. This Pockets clip was sort of this weird documentary where people were like taking stuff out of their pockets and showing it. It's kind of bizarre. But yeah, so, and then this sort of, you know, brings up a natural question of, well, can we identify something about the clip content itself that relates to prediction accuracy? And so we just sort of did this pretty brute force way of quantifying. It seemed to us, I guess I should say, from looking at this earlier pattern of results, that like some of these clips are sort of the most socially driven. So there's just like a lot of characters on screen. There's a lot of dialogue. There's sort of, I mean, these are short clips, so you're not getting like a full picture of the plot, but there's sort of like a lot of intrigue and a lot of like human factors going on. And so basically to try to quantify the overall level of social content in these movies, we just extracted the number of TRs that contained at least one face as kind of a proxy for how much social content there was in each clip. And it turns out that there was a strongly positive relationship between that and prediction accuracy for this cognitive score. So in other words, the clips with more social content did better for prediction. And this is kind of interesting because, I mean, this is the cognitive score. I mean, there's nothing, you know, in particular about this that is measuring social abilities. And in fact, the two factors were not super highly correlated. So the cognitive measure and the social measure, but still the clips with more social content are doing better for predicting this cognitive score. And just to show you some stills from these, you can see these clips have a lot of humans, a lot of dialogue. The clips that did the worst were sort of these like nature montages that didn't have a lot of humans or dialogue. And again, we tried to, and I think mostly succeeded in ruling out sort of low level arousal confounds from this as best we could. And I'm gonna, I'll go through this real quick, sorry, I'm almost done. So one thing that I think gets asked a lot, rightfully so, and that we ask ourselves when dealing with naturalistic movie watching data is how much of this is just sort of a function of where people are looking. So are there differences in basically where people are directing their gaze that could be explaining some of these differences in brain activity and therefore connectivity? And so we did actually have in this data set, they did collect eye tracking. It wasn't the highest quality in all cases, but we were able to get some usable eye tracking data as people watch these movies. And we hypothesized that, so I guess going back to this real quick, so we thought it might be the case that social content might just be more engaging overall, right? And if that's true, it might be driving people's attention in more similar ways. And maybe we could broadly index that by how similar people's gaze trajectories were. So if everyone is sort of tends to be looking in the same place on the screen, that's kind of what the director wants, right? They want to be sort of leading your attention through the scene. And maybe we can use the eye tracking data as sort of this like rough proxy for how engaged people were in the clip. And so if we then hypothesize that more engagement would lead to better prediction just based on sort of arousal or the ability to sort of corral people into a similar brain state, which makes our model better, then we would hypothesize that clips that evoke more synchrony in gaze trajectory should also be better at predicting cognitive ability. But in brief, that's not actually what we saw. So there was no relationship between the median synchrony of gaze in a given clip and that clip's prediction power. But there was, interestingly, a relationship between the standard deviation of people's gaze. And so what that means is in certain clips, there were pairs of subjects that had a very similar gaze trajectory, and then there were other pairs of subjects that would have very different gaze trajectories. So basically the standard deviation across people in terms of what they were looking at and when, the more deviation there was in a given clip, the better that clip did for predicting this cognitive score. So I think what that starts to suggest is that it's not necessarily like overall levels of engagement across people, but it's that people might be engaging in different ways with the clip, and that might be related in some broad way to this measure of cognitive ability. But you know, this was all pretty exploratory. This data set was not necessarily set up to answer these questions. So actually one of the biggest weaknesses here is that there's not actually that much variability in the clips. So most of the clips come from these sort of fast-paced Hollywood films and things like that. So we weren't able to, you know, really drill down into this as much as we had hoped. But, you know, I personally think this is kind of an interesting thing that I'd like to follow up on and hopefully acquire some more simultaneous fMRI eye tracking and, you know, see how the individual gaze trajectories relate to both brain activity and also behavioral traits. So I've already gone longer than I promised, so I'll just sum up here. Individual connectomes show some degree of stability, but at least in my opinion, I think optimizing for reliability in and of itself is not necessarily the right target. I think we should be optimizing directly for behavior prediction. We're seeing that using tasks over rest can in some cases dramatically improve sensitivity to behavior differences. And in my unbiased opinion, I think naturalistic tasks are particularly promising candidates to sort of draw out this variation, not only in ways that are more powerful and more sensitive, but also in ways that let us relate it back to the stimulus itself and sort of pin some of the differences we're seeing to differences in content in ways that I think can get us more traction and more interpretability than what we typically have during rest. So yeah, I'll stop there, and thanks for putting up with me this long. I'm happy to take questions on this second part.
Thanks, Emily. All right. Yeah, you know, you're in trouble for going overtime. You promised an hour and a half, and so an hour 40. I don't know. And that was really great. It's you did my my introduction. You know, gave me quite a bit of credit there because you just demonstrated some really awesome stuff with drilling down into the precision with the signal and trying to think about mechanisms down at that kind of biological level, but also really zooming out to these much bigger questions that I think are hard for us to grapple with, but are important if we ever want to use things like imaging for clinical benefits or anything like that, is to work out just what parts of the signal should we be tapping into and what kind of context should we put people into in order to find whether or not they're on the wrong track towards some pathology. We can jump in early. So I think all this stuff is really, really great to see. And I think it's really great for the students as well to sort of see the way that you guys have gone in as well, trying to work out ways to rule out different ways in which the signal could have been driving, or some of the things that could have been driving the effects, right? Like trying to rule out the eye movements, trying to rule out different contexts in different elements of the movies, I think is a really great way to kind of really drill down into these things. So that was really great to watch. Do we have any questions for anyone? Feel free, there's just a small group of us, so you can unmute and show your faces if you'd like to ask a question. Joe, you are muted. You got anything you'd like to ask?
Yeah, I've got I guess more technical questions later, but just on the last piece, that was fascinating, and just sort of riffing on something Max said about how, you know, you're starting to try and tease apart what exactly was it, and you were looking at you know, the number of TRs where faces were in. You were looking at the variability and so on. Something I was curious about was, you know, whether you could tease apart the visual versus the audio stimulus. You kind of observed how in some of those movies that we saw big differences in, like Ocean's 11, you know, the dialogue is very complex. You know, that's one thing that's always stood out to me about Ocean's 11, that the dialogue's really interesting and there's so much going on. Not that I'm suggesting you would need to have done this, but I wonder, what are your thoughts on what would happen if you only showed them the video or only showed them the audio, or is it the synergy between them that's really, you know, giving the big differences here?
Yeah, that's a really, really good question. We did I'll just share this again real quick, since this was my one hidden slide that you asked about, so I'll take advantage of having, yeah, I mean, it doesn't get at the problem that, yeah, or I don't know if it's a problem or a good thing. I mean, so dialogue is just, you know, something that we're obviously like really, really attuned to as humans, and it's a huge part of social information in the social world. We did also, in these particular clips, which again, you know, were not collected for this purpose, so they weren't sort of like ideally we would have had kind of we would have had something like exactly what you're saying, so like, you know, a clip with and without dialogue, or things that we're sort of deliberately manipulating or selecting clips the best that we can, you know, given that there's so many features that go into them. So, you know, we're sort of limited in the inferences we can draw from this particular set of clips. But one thing that we did look at that sort of gets at your question is these low-level audiovisual features. So we also extracted for each clip both the mean and the standard deviation of these kind of low-level physical features. So just brightness or luminance was the first one, optical flow, which gets at motion between frames, and then this audio RMS, which is loosely sort of a measure of volume or volume change. And then we correlated those all across clips with the prediction accuracy for the cognitive trait. And the only one, there were some, these are, I guess, the grayed out ones don't withstand correction for multiple comparisons, but there was actually a really strong relationship between the standard deviation of the luminance. So basically how much a clip is going from light to dark throughout the clip, and prediction accuracy, but it was in the inverse direction. So basically clips that are sort of more stable in their luminance are better for prediction. I think one of the difficulties in interpreting that in this particular set of clips is the clips, so like the especially the Ocean's 11 clip, it's like really dark, so they're kind of in this room at night and they're having this conversation and there's just not that much light. So, you know, if you have a low mean luminance, you're also going to have a low standard deviation of luminance, I suppose, if the whole thing is just sort of like on the darker side. But, you know, whether that sort of made them like lean in almost and like try to, you know, they're not like necessarily getting hit over the head with like the sensory, you know, it's not some big like flashing action chase scene or whatever. It's sort of like, it's, in this case was like sort of unfortunately inextricably linked to the social content, because it was like those clips that were more social in nature also just tended to be like shot in darker light. So, I mean, I guess you could flip that on its head and say maybe the most important thing is not having a lot of light, but my bet would be that it's more in the social content. But I think one of the things we'd really like to do at some point is actually, you know, redo something like this with a set of clips that are a little bit more deliberately selected to vary on these things. And I mean, I think the last part of your question hits us something really interesting that I've been wanting to do for a while, which is, like so I've also done some work with purely auditory stimuli. And I actually think in some ways, like just narratives where people just listen to a narrative and there is no visual input. And I think in some ways that actually leaves more open to interpretation, because you're kind of engaging, like as you're hearing a story, you're kind of like engaging in visual imagery, and that imagery that you're engaging in, that could differ across people more than like if you're watching a movie. And this is actually one of the like bigger challenges that I've been finding in my work, not on these publicly available data sets where the stimuli are chosen for me. I think it's actually really hard to find stimuli that are truly ambiguous, because usually the director or the creator, whoever it is, sort of has a preferred interpretation that they're kind of trying to guide you towards. But I think the idea of using silent films, like either films that were designed to be silent, or by stripping audio away from the films, or conversely using auditory only information and sort of a story where people are encouraged to engage in their own imagery of what's going on. That's something like that, my hunch or my gut feeling would be like that could actually get us even more sensitivity to these individual differences, because it's sort of constraining everyone in a certain way, but not so much, and still leaving room for that individual variability to come into play.
Cool. Thanks.
Mac, just a quick comment. Can I go ahead?
Yeah, go for it, Louis.
Yeah, so Emily, one thing that we're finding, I mean, we're finding the same movies have the best prediction too, in a completely different way of doing it. And the funny thing is, and we don't know if this is meaningful at all at this point, but we haven't done careful statistics with this, but what we find is that because we do these kinds of predictions temporally, as basically a sliding window on the movie, we find that certain segments of Ocean's 11 and other movies seem to be particularly good. So, I mean, this is an impression at this point, is nothing more than that, but it really doesn't seem to be, it actually seems to fluctuate quite a bit across the movie. So to the extent that that's actually something that we can make solid and really do some statistics and some analysis, it leaves open the possibility that there are certain segments of clips or movies or stories that could be most predictive about certain aspects of individual differences, right? So there's the spatial temporal, if you will, element of this whole equation that is also, I think, could be quite interesting. Although it opens a lot of challenges, because what does it mean for a segment of 10 seconds to be really predictive of IQ and some other, I mean, it starts getting a little messy, right? I mean, it's a little bit too non-hypothesis driven in a sense, or too exploratory, but yeah.
Yeah, that's fascinating. I saw the preprint from your group. I haven't done a deep read, but I did sort of scroll through, and I was heartened to see that we did sort of converge on like these same clips, which is really cool, and certainly makes me more confident in these results here. And I think it's fascinating to look at the temporal element. I mean, that's kind of where I have another data set that we're working with where the movies are a little bit longer. It's actually data I collected myself for once, and so but I mean, I think the tension that you identified is absolutely, I feel that all the time. It's sort of, I always kind of want to get down into the dynamics, but then I sort of, I worry about being able to like interpret, I don't know. Yeah, it just sort of blows up at that level, but I absolutely believe, I mean, I think it makes sense that there would be, specific moments that are driving this more than others. And the question is just sort of, how do we go about identifying those in a rigorous way and like a generalizable way? The other thing is like, we don't necessarily want everything we find to be only true of that one stimulus, or maybe that's sufficient for certain purposes. Like if we find like the movie that can diagnose schizophrenia, then I guess we don't care what, but ideally we would be able to extract
yeah.
more general principles, and the movies are so different on so many different parameters that it's super
But I
challenging.
think it's fascinating if we had larger data sets, right? We can actually use some part of the data set to identify some of these segments, these blocks that seem to be particularly informative, and test it completely out of sample or some other sample or something like that. That I think that's what would be really needed to be a little bit more confident that we're not just, I don't know, whatever. I mean, something stopped during the scan who knows? I mean,
totally. Yeah. I think another idea I've kind of been batting around with some collaborators is like, could we kind of like splice together like a super movie that we think is, comprised of these things that, I think it's a little early days for that, but I think I think the sort of data sharing aspect of all this and the fact that the naturalistic tests are becoming more popular, I think that, we will be able to hopefully move in that direction. And I guess the big cognitive, well, one of the many cognitive questions that I would have about that would be like, even if you are seeing that, like, for example, this particular 10-second period is somehow really diagnostic or really stratifying, like, do you still have to have the context leading up to that in order to, like how much of that is sort of a consequence of like having seen something and being involved in a plot? If you did sort of just splice that out and look at it on its own, would it have the same effect? And I mean, the answer to that probably depends on the stimulus, and it might be different for different stimuli. But I think it's also really interesting to think about like temporal dynamics in the sense of like context and the idea of like Ori-san's beautiful work on like temporal receptive windows and stuff like that. And so there's just like a ton to do, but it's cool. I've been following the work from your group too. So it's really cool to see at least some convergence, which definitely makes me trust my stuff more. So that's good.
So Emily, Claire had a question, and then maybe we should wrap the session up because I'm worried that you're up late trying to walk us through stuff while we're sitting here in the middle of the day. So maybe we'll make this the last question and then we'll say thank you.
I'll make it a quick question. Hey, Emily, nice to see you.
Hi, Claire.
Again.
It's so nice to see you.
I know. I think the last time we saw each other was in Cambridge over gin and scotch?
Yeah, I have such fond memories of that. So Claire like took pity on me because I was in Cambridge by myself. She took me out and showed me a great time, and I still hope to return time when the world gets back to normal. So anyway, nice to see you.
Yeah, I brought her to a very good gin place.
It was glorious.
Yeah, thanks. This talk was really awesome. You covered so much cool stuff. I just had a question. I think about this often, and your talk really makes me think about it. The extent to which, on the one hand, we're, developing these really amazing ways of, looking at individual connectomes, and, we develop quite sophisticated cognitive tasks and that sort of thing. But we still sort of measure behavior out in the world in a pretty clunky way, right? We'll have, this beautiful complex sort of, cognitive task and imaging, but then we'll measure behavior, using these outdated questionnaires that will ask things like, do you fidget in lectures, or do you enjoy going on roller coasters, right? And what I kind of wondered, I think it's so amazing how you're seeing this predictive ability with the naturalistic movies, but thinking about, say, something like rest. I mean, I think what's going on in the brain and people's sort of phenomenology in rest, I think that would have implications for how they are out in the world, right? I don't think it would necessarily relate to some of the kind of course behavioral questionnaires or, IQ measures, but do you think there's, part of it is that we just haven't paid enough attention to sort of measuring behavior out in the world in a more rich way, and we tend to measure it in a way that kind of ignores a lot of the nuances and actually converges people together, particularly if they're healthy people filling in a Likert scale with 4 options. And I wonder if we could imagine a really nuanced measure of behavior out in the world, looking at sort of maybe how people, how much people mind wander, or how like engaged they get in different topics and stuff, and that something like rest would actually predict that really well.
Totally. Yeah. Awesome point. I completely agree. I feel like there's so much irony to like the fact that we have these like super sophisticated brain imaging things, and the things we're trying to predict are like these answers to these. And it's I don't know, I think about it all the time, and I don't have a great answer, but I think it's true, like what you say. I mean, one of the biggest challenges to studying individual variability in the healthy population, which I didn't go through my whole background spiel on this talk, but I do think one of the points of that is that, you know, a lot of the clinical phenotypes are sort of the extreme ends of things that tend to be more dimensional, even in the healthy population. So, you know, I am undertaking this work with the hope and with the goal that, you know, by sort of parameterizing things, even in a more normative sample, that will lead us to better understand what happens at the extremes. But as you point out, I mean, a lot of the behavioral instruments we have are pretty coarse in, you know, healthy typical adults. They're like, there's a sense that we're not, you know, capturing these more subtle differences that actually may be really important. You know, I think some reasons to be optimistic, not based on anything I'm currently doing in my own lab, although hopefully we'll get here at some point, like some of the like mobile phenotyping. I mean, there's privacy and ethical concerns, but I think being able to harvest data from smartphones about, there's all sorts of things. I mean, you know, like geographic roaming entropy was one thing that there was a cool paper out on that recently from Kate Hartley and Aaron Heller, where I think they were able to predict it was a specific seed-based connection in the brain, but it was at rest, and they were able to predict basically how much roaming entropy people has. In other words, like how much geographic diversity there was in places that they had visited over the last couple of weeks. So that's like something that's so much more ecologically valid, and also known to be related in some cases to like other risk factors for, you know, depressed people just tend to move about in the world less, you know, and like, or even like, you know, without getting to the clinical side, just like some sort of, you could extract some kind of like personality, you know, like, I don't know, just like openness or energy or whatever. And then that's not even getting to like how people use their phones to talk to other people. So pulling out like sentiment of text, or even just frequency of text, like how often are people engaging socially and how many social partners are they connecting to on a given day. And like, I just feel like that kind of stuff is so much richer and more ecologically valid. And, you know, it's harder, rightfully so, to get that, like because of privacy and things like that. But that's the kind of thing that kind of gives me more hope for this, to be honest with you, like at the end of the day.
Yeah.
So yeah.
No, that's awesome. And luckily we've got people like Mark Zuckerberg, you know, make it easy for us to get at all that information.
Yeah, I know. I feel like kind of dirty saying it, but I honestly think it's like really interesting.
I mean, it's
I
sad.
think the other
Kind of cool as well.
it's
Anyway,
really,
thank you. Yeah, that's an awesome point. Thank you.
yeah, but I think too, like some of the work that I know some of you guys have done too, like with like more computational phenotyping, so trying to, you know, derive like more latent factors from I guess hypothesis driven is the right word, but just sort of more incisive ways of, you know, designing tasks that are just better and more interesting and like more, you know, more easily parameterizable in sort of computational terms, and you know, using models to tease out these latent factors that might be more variable across, you know, otherwise healthy people. I think there's a lot of promise there too. So it's something I'm thinking a lot about. You know, I think in a lot of our past work, we've sort of been limited by the measures that exist in these large scale data sets, which, I mean, not to criticize, I mean, it's amazing that this data is out there and available. And I think when people collect this data, they have to make hard choices, and it's hard to push for including these like, you know, sort of less well trodden or like unproven types of behaviors. But I think people are sort of realizing this more and more, and there's some really excellent work out there that hopefully our behaviors will sort of catch up in sophistication to our brain imaging measures. But
Awesome. Thank you.
Cool. Well, great question and answer to end excellent session. Thank you again so much, Emily, for joining. Real pleasure. And I'm really excited to see what you come up with, right? As we get better measures and better of behavior and neuroimaging, let's see, you know, what you can find. I'm really looking forward to watching. So thanks very much.
yeah, thank you so much for having me and for sticking around this long. It's been a pleasure.
Awesome.
Thanks.